Welcome everyone to the Precision Medicine Seminar. Today we have a guest speaker from Canada, Dr. Brent Richards, who is an associate professor of medicine with tenure at McGill University in Montreal. He's actually a clinical research scholar and a physician scientist associated with 3 different departments at McGill: Department of Medicine, Department of Human Genetics, and Department of Biostats and Epidemiology. He's also an honorary senior lecturer at the King's College in London. Dr. Richards is a quintessential physician scientist. He is trained in medicine, earned his MD from the University of Calgary in Canada, and then he completed his residency and fellowship in endocrinology at McGill. He then earned masters in epidemiology and biostats at McGill as well, and did his postdoc in genetic epidemiology at the Twin Research Unit at the King's College in London. He currently practices endocrinology at McGill and also runs a research program in genetics and precision medicine. And his research program is focused on the identification of genetic determinants of common traits, common complex traits with special focus on endocrine traits, and also translation of these findings to improved clinical care. He has led a number of genetic discovery studies published in Nature Genetics. He published over 120 papers. He's an elected member of the Royal College of Physicians and Surgeons of Canada, and also the American Society of Clinical Investigation. More recently, I had a pleasure to collaborate with Brent on within the global COVID-19 host genetic initiative, where he took a leadership role in the exome and genome sequencing work group, addressing the role of rare genetic variation in susceptibility to COVID-19 infection and its complications. And I think he will discuss some of this work with us today. So welcome, Brent, and thank you again for taking time to visit us virtually.
Christoph, it's a real pleasure, and I thank you for the invitation. This has been a very unusual couple of years for all of us, and it certainly has been so for me, because as Christoph said, I'm an endocrinologist and therefore don't know very much about infectious disease. But at the time of the beginning of the pandemic, we were in the process of creating an institution-wide biobank, similar to what you have done at Columbia. And we were asked by our funders to actually orient that towards COVID research. And so what I'm going to talk to you today is about some of the research that has come from this, which is entitled The Influence of OAS1 on COVID-19 Outcomes, Neanderthals to the Rescue. I'm just going to add as a side note, one of the really fun parts of this whole adventure has been meeting people like Gundula and Christoph, who have been really helpful in helping us to gain some insights into what predisposes some people to severity of disease, and really learning from them through this journey. So thank you very much, Christoph, for inviting me. I'm going to start off with my conflicts of interest slide. I should let you know that I'm more Neanderthal than almost all of you. That wouldn't be a surprise if you happen to know me, but this is what 23andMe claims. And I also have founded a small biotech company called 5' Sciences. I wouldn't have really been very comfortable starting this adventure without the support of these 3 people who really led a lot of our COVID research. This is Suri Zhu, who has recently started as an assistant professor at McGill University in Human Genetics, who's a very capable postdoctoral fellow in my lab at the time and led this program. Guillaume Butler-Laporte, who was doing research in our lab and is an infectious disease specialist. As well, we were very lucky to, prior to the pandemic, to have Tomoko Nakanishi join our group, who is a respiratory physician from Japan. And so with support of these 3 people, as well as an enormous number of people who collected data and samples from across our institutions, we were able to try to use some of the tools that we had developed some comfort with over the past 15 years to try to understand how this little guy was influencing severity of disease. I'm going to start off by just talking a little bit about this data resource that we've created that you can apply to and get access to data for. It's called the biobank québécoise de la COVID-19, or the Quebec biobank for COVID-19. We've collected data and samples from 3,431 patients, a large number of whom have severe COVID-19, many of whom had mild disease. And we've completed over 8,000 visits on these people. You can have all of this data. You can have you can have many of the samples if you provide justification. You don't need much justification for the data, but you do for the samples. And there's lots of data. So we've undertaken proteomic analyses on about 2,000 of these people. The whole genome sequencing is actually now done on over 2,000 individuals. The genome-wide genotyping is now done on everybody. The whole genome sequencing is approaching 2,000 individuals now. We have transcriptomics, which will be done on 2,000 individuals, immunoserology, as well as metabolomics from Metabolon, standardized core laboratories and immunoinflammatory markers, and then all of their clinical information that was captured during their visits to the hospital. We have lots of controls, which is very helpful for a lot of research programs. And you can apply for access to this data. You can just type in bqc19.ca, and really, as long as you respect the confidentiality of the data and pay the nominal application fee, then this data can be transferred to you. So yours for the taking. I'm going to tell you a story about how we've moved one target, OAS1, from a genetic association to drug development in 12 months. And that has been really accelerated in the time of COVID research, when a lot of money and resources, for obvious reasons, have been dedicated to better understanding why some people have severe disease and other people do not. I'm going to structure my talk around these 7 different bullets. The first is, I'm going to ask the question, can disease influencing circulating proteins be identified without model systems? I'm then going to talk about the genetic determinants of COVID-19 severity. I'll then zoom in on OAS1, which I'll spend most of the talk explaining. I'm going to talk about the contribution of Neanderthals to our pandemic, the contribution of bats, the work that we've done on some cool tool compounds for OAS1, and then wrap up with some conclusions. So, can disease influencing circulating proteins be identified without model systems? So, this would be very helpful, because if you can do so, and these are causal, then you may have a set of privileged drug targets, because circulating proteins are more accessible for drug binding. They are often signaling molecules. And in fact, they are the whole field of endocrinology, which is what I spend most of my clinical time doing. The problem is teasing consequence from cause, and in trying to identify the proteins that actually influence COVID-19 severity. And to try to solve this problem, we've used a study design called Mendelian randomization. Mendelian randomization is a methodology that is a type of causal inference. So that means that you explicitly aim to infer causality. And you do so by questioning whether or not some exposure, which in this case is a protein level, is causing some disease. And the way that these sorts of studies are done is that you are aware that most relationships between a protein level and a disease are subject to confounding. And that confounding can bias those causal estimates. Furthermore, the disease itself can influence the level of the protein through reverse causation. And this can also lead to biased estimates of the causal role of any exposure, but in this case, protein levels, upon the disease outcome. And so you can use human genetics to disentangle this problem of confounding and reverse causation, because if you have genetic variants that influence the level of the protein, because of Mendel's second law, which, to remind you, is the random assortment of alleles at conception, you break any association with confounders in the population, with one exception of population stratification, which I can go into in detail if you have questions. But this is very powerful, because what you can actually do is you can deconfound the relationship between your exposure and your outcome. It can almost never be influenced by reverse causation, because reverse causation, of course, happens when the disease itself influences the level of the protein. And because the genetic variants are assigned to the person always before the onset of any disease, such study design is not subject to reverse causation. The main problem with the study design is that if the genetic variant influences the disease through a pathway independent of the exposure, where the exposure is the protein level, then you can end up with a biased estimate. And this pleiotropic pathway is what we spend most of our time in Mendelian randomization trying to avoid. And in the case of circulating proteins, there may be a way that we can look at this study design by reducing pleiotropic pathways. And this can be done by using what are called cis-pQTLs. So what are cis-pQTLs? Cis means close, P means protein, and QTL is a quantitative trait locus. So all this means is that if you do a genome-wide association study, which is to look across the genome to understand which genetic variants are associated with something like a protein level, and you identify genetic variants that sit right beside the gene that encodes the protein, or in fact are in the gene that encodes the protein, or in fact are in a promoter or enhancer of that gene, then it is very likely that these genetic variants act upon the gene itself, influencing its transcription or translation to influence the level of the protein. And if that is the case, and you can identify cis-pQTLs that are influencing protein level by being very close to the gene that encodes that protein, then you can hope to reduce some of this pleiotropic pathway, because it's very unlikely that that genetic variant is influencing the disease in a way that is independent or not acting through the protein level. And so we've used the study design for many different diseases, and because we had some familiarity with it as well as its problems, we decided to point the lens of this program where the disease is COVID-19 severity. And so to do so, what you need to be able to understand is the genetic determinants of COVID-19 severity. And so with Christophe and Gundula, as well as many other, literally hundreds of other people around the world, what we decided to do through leadership from Andrea Gana, as well as Mark Daly, who are both at the Broad Institute and in Helsinki, is we pooled our data with a common outcome definition, common control definition, to identify areas of the genome which are strongly associated with COVID-19 outcomes. And this was done through a genome-wide association study. This is now published in Nature. And what you can identify through this Manhattan plot, where, to remind you, the y-axis is the negative log 10 p-value, in the x-axis is genomic location, is very clear signals that surprisingly arise from the genome and are associated with COVID-19 severity. And so you can see that there's this huge signal at the chromosome 3 locus, and we have really, at least I really have no idea what's going on here, despite spending a lot of time on this locus. We decided to focus our energies on this one, which is called OAS1. This OAS1 is a circulating protein, and therefore we can apply the study design of circulating protein Mendelian randomization studies. And so now I'm going to talk about OAS1, and I'm going to talk about how we came to believe that this protein may be influencing COVID-19 outcomes. So to remind you of the study design, we have taken genetic variants that are associated with the level of OAS1 in people who are not infected. And I'm going to say that again because it's really important, and I'm going to come back to it several times during the talk. So this is genetic variants which are associated with the level of OAS1 in a non-infected state. They may not be genetic variants that are associated with OAS1 levels in an infected state. And that's really important to keep in mind, and I'll come back to that. We're blocking this confounding path because of Mendel's second law, and we hope that we're reducing this potential pleiotropic path because the genetic variants that influence OAS1 actually sit inside OAS1 in one of its introns. And now what we're going to do is simply ask the question, does genetic variation controlling OAS1 levels influence COVID-19 outcomes? And so this is work which we have published, and I would encourage you, if this is of interest to you, to take a look at this work where I will go over some of the high-level details, but would be very happy to dive into any of the technical details if people have questions. So when we applied the study design, the question that we were able to ask is the non-infected OAS1 level effects on COVID-19. And we used 3 different outcomes. The first is susceptibility to COVID-19. The second was hospitalization for COVID-19, and very severe COVID-19, which was ventilatory assistance, death, or visit to the ICU. And what you can see is that per standard deviation increase in the log of OAS1 levels, we found that individuals who had higher OAS1 levels were protected from COVID-19 susceptibility. In fact, the odds ratio is 0.78. The p-value is quite low, and the confidence intervals therefore excluded the null and were fairly tight around 0.8. When we looked at the hospitalization outcome, we saw a stronger effect such that the odds ratio is 0.68. The p-value was quite low, and the confidence intervals were rather tight. And then a further decrease in the odds of very severe COVID-19 when we looked at this as an outcome where the odds ratio is 0.54. And so I told you that the genetic variants that control OAS1 levels in the non-infected state reside inside OAS1. And so we took a deep dive to try to understand what could be going on here. There's actually been a lot of work done on OAS1 by different groups around the world, but predominantly one from the University of Chicago, Luis Barrero, who actually used to be in Montreal, and so I knew of his work. And what Luis has been able to show is that one of the strongest determinants of any isoform in the entire genome, where, to remind you, isoforms are different forms of a protein, which include different exons, is this SNP here, which leads to the inclusion of the 6th exon in this protein called OAS1, which is very interesting that one of the strongest, so this is a splice variant, and this is one of the strongest splice variants in the human genome, which was actually first described by my colleagues at McGill University in the early 2000s when they tried to identify splicing variants across the human genome, published in Nature Genetics by Jacek Majewski. And it is actually this exact same genetic variant which is associated with circulating OAS1 levels and is very strongly associated with severe COVID-19 outcomes. And you can see that there's actually different isoforms of OAS1. P46 is the one which is protective, which is due to the inclusion of this G variant at RS10774671. And the P42 is by far the second most common isoform. And so therefore we started to ask, well, this is interesting. Are we actually looking at a splice effect or are we looking at an expression effect? And because we have both splicing and expression data for OAS1 and non-infected individuals, we can ask which one is affording the protection against COVID-19 severity. And the results were pretty clear. What we found was that the expression effects were all diminished so that on the same exposure, meaning a unit increase in the log level of exposure, there was no clear susceptibility effect, no clear hospitalization effect, and a rather weak protection statistically against very severe disease. But when we looked at splicing, we found a very different story. We found that the splicing effects were very strong with odds ratios very low, confidence intervals very clearly excluding the null, and these effects were stronger in hospitalization and stronger still in very severe disease, suggesting that the biological mechanism whereby people are afforded protection against severe COVID-19 outcome does not depend upon the amount of COVID-19 of OAS1 expressed, but the type of OAS1 expressed through the splicing SNP present at the 6th exon, which leads to the P46 isoform. Now, I'm going to get into a little bit of a technical detail, which is really relevant to those of you who work in this field. But you can also confuse yourself in these sorts of studies when you have what's called confounding by linkage disequilibrium. And that can happen because so many different diseases are polygenic, and you could just have two separate biological signals influencing an outcome to both the exposure and the outcome at the same locus. And those two signals are only linked to each other through linkage disequilibrium. And so it's very important that you take a look at colocalization to understand if you are observing the same signal for both the exposure and the outcome. And that is indeed exactly what we find. So at OAS1, this is the association with OAS1 level. and this is the association with severe COVID-19. You can see that the signal arises from the exact same genomic location, but it's a very high linkage disequilibrium, suggesting that this is not confounding by linkage disequilibrium, but really is underpinned by the same single genetic signal. And so that allowed us to get somewhat excited about the effect of OAS1, but we really wanted to drill down on to whether or not the splice variant was actually the causal SNP at the locus. And so this is actually published now. This is from Jennifer Huffman in Nature Genetics. It was published this year. And the problem with discerning the causal SNP at this locus is that if you look in you people of European ancestry, and I'm going to get back to this in a minute because it becomes very important when I'm going to talk to you about Neanderthals, is that the linkage disequilibrium across this locus is very strong. So there's a very large linkage disequilibrium block, and that's due to the fact that in Europeans that it's from Neanderthals. And so it's very difficult to disentangle if the lead SNP, this is the splice site variant, is the causal SNP. And so what we did was we turned to another ancestral population, which is people of African ancestry, who have a very different linkage disequilibrium picture at this locus, where actually you can vary this splice site variant with having very little carriers, meaning very little covariance from the adjacent SNPs by breaking up the linkage disequilibrium block. And if you ask whether or not this SNP actually has an effect upon the outcome, and these ones don't, this would then lead you to have more confidence that it was this single genetic variant that was driving this signal. And so that's exactly what we did. And these, we looked at people of African ancestry, and you can see that the effect of this genetic variant is very similar to the European effect. Here's the European effect here. That when we meta-analyze this across people of African ancestry, we find almost the exact same effect size. Confidence intervals are a little bit wider because there's many fewer people. There's 2,700 cases versus 17,000 Europeans. But this really underlines the importance of initiatives such as yours in Colombia, where you're collecting a large proportion of non-European genomes, which allows us to be able to disentangle what could be the causal SNP at this locus. And it seems that the driver of this association is indeed just the splice site variant at OAS1. And so if we now believe that there is a role for OAS1, we can start asking questions of what is this thing? So OAS1 and my clinical colleagues in virology were not at all surprised to find that OAS1 is playing a role within host to SARS-CoV-2 infection, because as you know, the SARS-CoV-2 virus is a double-stranded RNA virus. And one of our mechanisms as humans that allows us to chew up double-stranded RNA is through activation of this protein called RNase L. And RNase L is actually activated by OAS1. And so by turning on OAS1, you actually lead to higher degradation of double-stranded RNA from viruses, providing a clear mechanism whereby OAS1 would protect against severe COVID-19 outcomes. It's really important to mention that it's interferon stimulated, and I'm going to talk about that in a moment. And that the same SNP, which we identified to be protective against severe COVID-19 outcomes, actually influences response to other viral infections. And so it is the same allele protects against other viral infections, such as West Nile virus. And I'm going to come back to this at the end of the talk, but there are actually two drug classes that are known to influence OAS1 levels. And I'll show you some of the work that we've been doing on very early drug development using this type of compounds called PDE12 inhibitors. So we have reason to believe that OAS1 is important, but we're still skeptical because sometimes when you look at a problem from one angle, it becomes very different when you look at it from another. And so we actually, instead of using human genetics, we turn to observational epidemiology to try to ask the question, do people who have higher uninfected levels of OAS1, are they afforded protection against severe COVID-19 outcomes? And the corollary to this is what happens to OAS1 levels during infection. But the problem with that is that OAS1 is interferon stimulated and therefore is highly expressed in people with severe disease. And so the disease itself actually increases OAS1 levels, leading to the problem that we tried to avoid with the Mendelian randomization study design that I began the talk with, and that is reverse causation. And so what we would expect to see is that individuals who had higher baseline levels of OAS1 in a non-infected state were protected against severe COVID-19 outcomes, but that during infection, it may actually be reverse because severe disease would increase interferon stimulation, which would increase OAS1 level. And so what did we find? We found that non-infectious state individuals who we measured OAS1, higher OAS1 levels, so this is again per standard deviation increase in the log of OAS1 levels, we found estimates which were very similar to what we had found from the Mendelian randomization studies. But when you look at acutely infected people, you find the opposite. And that's because the infection itself changes OAS1 levels. Probably because the host is trying to chew up all this DNA. And in order to do that, it has to activate the OAS1 pathway. So what we have been able to establish so far is first that OAS1 levels are predicted in the non-infectious state to protect against severe COVID-19 outcomes, that this is driven by the P46 isoform of OAS1, and that is caused most likely by a single genetic variant, which increases the amount of the P46 isoform of OAS1. Okay, so now I'm going to tell you a story about Neanderthals, because this becomes very important in understanding how it is that humans came to use the OAS1 pathway to protect themselves against RNA viruses. And so not understanding anything about Neanderthals and learning that the OAS1 locus is actually derived in Europeans from Neanderthals, I reached out to somebody who spent most of his academic career looking at Neanderthal genomes. And this is Hugo Zeberg. He works at the Karolinska Institute. He's also a physician. He's an emergency room physician, and therefore the only emergency room physician I know who spends his academic time working on Neanderthal genomes. And Hugo, when I got him on a Zoom call, he said, well, yes, Brent, I can tell you exactly what's happening. What happened was that the P46 isoform, which is the protective form of OAS1, is very prevalent in sub-Saharan Africa. And when multiple migrations of Homo sapiens left the Rift Valley, the lazier ones, like my ancestors, stopped in Europe, and the more adventurous ones walked through Asia to the tip of Tierra del Fuego. And what's fascinating is that when Neanderthals left, approximately 60,000 years ago, they took with them this protective form of OAS1. But when modern Homo sapiens left the Rift Valley and migrated through to Europe, for some reason in the Middle East, they lost this genetic variant. So they lost the protective allele from P46. And so they were then very susceptible to pandemics and double-stranded RNA viruses. But then as they migrated into Northern Europe, they actually met with the Neanderthals and thankfully interbred with them. And the Neanderthals actually reintroduced this lost genetic variant into the European and Asian ancestral populations. And then an explosion happened. So this is the frequency of the OAS1 P46 allele going back in time from Eurasians, going back to the last glaciation where it was actually relatively rare, 40 to 20,000 years ago, where that increased to be very common, achieving an allele frequency of 30% in present times. So really an explosion of allele frequency, increasing approximately 10-fold. We don't know why, but likely because of the selective pressures from RNA viruses. And then, interestingly, allele frequencies stop at exactly 30% for reasons we don't understand, but are very stable across different non-sub-Saharan African populations, which suggests, at least to me, that there is some disadvantage of having this P46 allele, and that populations do not tolerate higher levels of P46, but freeze at about 30%, in fact, 32% of most populations, very consistent across South Asia, across East Asia, across Europe, with some more variation in the American continent. And so what this suggests is that it reminds us that our species has been under selective pressure from pandemics for millennia, and that these thousands of years in our evolutionary history have allowed us to provide some protection against SARS-CoV-2 in the present sense, because for Eurasians, we received the allele back from Neanderthals. And now I'm going to talk about bats, this is a work that has been done by Sam Wilson and his colleagues. So Sam Wilson has become a close collaborator since he published this paper in a preprint archive form. It was published in Science, and at the same time, Sam approached this problem by looking at the genes that were stimulated by the interferon system. And Sam Wilson is a virologist, and early on in the pandemic, he and his colleagues picked up that interferon-stimulated genes would be the kind of genes that the host uses to protect against SARS-CoV-2, because those interferon levels increased dramatically during SARS-CoV-2 infection. And so what Sam and his colleagues decided to do is to set out using entirely different tools, this time using cellular models in virology, to understand which interferon-stimulated genes had protective effects against severe COVID-19. And what they identified was OAS1. So they found, and this is the log 10 of the plaque-forming units. This is a way to be able to test replication of the SARS-CoV-2 virus within a cellular context. And what they found is that if you introduce OAS1 in the P46 isoform, so remember the human genetics predicted this is protective, and independently Sam and his colleagues at the same time predicted or observed that the P46 isoform dramatically decreased replication in their cellular model system. What they found also is that the P42 isoform, which is the other most common isoform in humans, had almost no protection against SARS-CoV-2 replication. And so why am I talking about bats? Well, the reason why I brought up bats is because Sam and his colleagues hypothesized that the reason why SARS-CoV-2 is so exquisitely sensitive to the P46 isoform is that bats don't have it. Explain this. So if you take mammals, from which most double-stranded RNA coronaviruses seem to have emerged and then jumped over to humans, so these are cows for the OC43 virus, the MERS virus, which is thought to arise from camels and bats. Both of these viruses evolved within 3 mammals, which have a functioning OAS1 system. Then they actually have the P46 isoform of OAS1. And so when they had the P46 isoform, this virus had to evolve under the selective pressures of the P46 isoform of OAS1 because it's present within these mammalian species, that when it jumped to humans, the virus was not too sensitive to it because it had already evolved in its presence, and it actually developed ways to fight against it. And now I want you to remember this because I'm going to come back to it when I talk about drug development. The way that the virus actually developed a mechanism to fight back against the P46 isoform is to develop a protein called PDE12. And that's a phosphodiesterase 12. And that is natural selection telling us the way to fight against P46 is to use PDE12. And that will come back when I talk about the use of PDE12 inhibitors in the prevention of severe COVID-19 severity. So here I'm talking about 2 viruses that are not SARS-CoV-2, OC43 and MERS. And now what happens in the SARS-CoV-2 pandemic is that it's thought that the 2 species from which the virus could have arisen are both rhinolohidae bats, which is a subspecies of bats, as well as perhaps pangolins. Now, neither of these two mammals, the subspecies of bat or pangolin, has the P46 isoform of OAS1. And what that means is that the SARS-CoV-2 virus has never had to evolve in the mammalian species under the pressure of P46. And this suggests that in a very sensitive mechanism to the that this virus would be very sensitive to P46 isoform of OAS1 because it never had to evolve in the presence of that virus, whereas these other two coronaviruses did. And so Sam actually tested this hypothesis. He took the OAS1 from Homo sapiens, as well as the OAS1 from the Rhinoholidae, which I'm probably mispronouncing. But you can see that this is the P46 of isoform of Homo sapien OAS1, which decreases replication profoundly. But if you use the OAS1 the OAS1 protein that comes from the Rhinoholidae virus, or Rhinoholidae bat, from which it is thought that the SARS-CoV-2 virus has evolved, you have no effect. And this suggests that the SARS-CoV-2 virus has never had to fight back against P46, suggesting that P46 may be a way to combat the virus, and that this could be, therefore, an Achilles heel of the COVID-19 pandemic. And so before I get into the drug development, I'm just going to summarize and walk you through what we have done so far. So really starting from work that was published in a preprint archive form as soon as the data was released, really through international collaboration, which was led by Andrea Gana. We had many different GWAS loci to dig into. We, through collaboration with Jenny Huffman, who is at the Million Veterans Program, as well as Hugo Sedberg, who I mentioned previously in Sweden, we identified what is a single likely causal SNP at the OAS1 locus. Suri Zhu then demonstrated the splicing effects upon severe COVID-19 outcomes. She demonstrated the effects of circulating OAS1 levels through pQTL Mendelian randomization studies, providing an effect estimate and a causal inference of that higher OAS1 levels in a non-infected state strongly protected against severe COVID-19 outcomes. This was again supported by observational epidemiology, which came to the same conclusions, as well as in vitro validation, entirely independently done at essentially the same time, unbeknownst to each other, by Wickenhagen et al. And published in Science. What I'm now going to talk about is some of the work that we've done using different tool compounds. And so I mentioned that the way that other coronaviruses have fought back against P46 isoform of OAS1 is to develop an inhibitor of OAS1 called PDE12. And that inhibitor is endogenous to humans as well. And so we thought that if we inhibit PDE12, that we can actually inhibit the inhibitor of OAS1 and therefore increase OAS1 levels. And so it turns out that longstanding collaborators at GSK had actually been working on this hypothesis previously, prior to the pandemic, when they developed a PDE12 inhibitor. And thankfully, they published this molecule. And so what we have done together with Sam Wilson in Glasgow is, in collaboration with GSK, is we have actually purchased the compound, and we are now testing the effect of that compound on SARS-CoV-2 replication within the model systems that Sam has developed in Glasgow, but also in an organoid system of an air-lung interface developed by NSR Lehman in northern Sweden. And what these studies will do, we hope, is provide an opportunity to directly test the hypothesis whether or not pharmacological inhibition, which inhibits the inhibitor of OAS1, where that inhibitor is PDE12, leads to a reduction in SARS-CoV-2 replication. And so those studies are ongoing. We're very thankful to have the support of GSK to do so. And the reason why they are interested is because we don't actually probably need more molecules to fight against SARS-CoV-2. viruses. But what we do need is general purpose pharmacology, which can be used against other double-stranded RNA viruses and perhaps pandemics of the future. And so with the hope that you'll have some questions and we can have some conversation around this topic, I'll end here. But prior to doing so, I'll summarize. The first point that I've tried to make is that increased levels of OAS1 protects against COVID-19 susceptibility, hospitalization, death, and ventilation. These results survive multiple sensitivity tests. OAS1 has clear biological rationale. Natural selection points to PDE as the key enzyme to inactivate OAS1. And compounds exist to increase OAS1 levels and therefore being tested for their inhibition of SARS-CoV-2 replication. I'm going to thank a lot of people here. This was only possible to get all this done in less than a year by the concerted effort of really dozens of people at my institution, but actually hundreds of people around the world sharing their data freely through an open consortium. I'm going to thank Vincent Moser, who secured the funding and led the BQC19, Daniel Kaufman, who led one of the other centers at another hospital in Montreal, but was also is also thankfully a virologist and was very helpful in putting together and making sense of all of these findings. I'm going to just briefly thank David Morrison, who ran all of the clinical collection here at my hospital, as well as thank funding partners, which included the BQC19 itself, the foundation at my hospital, the Public Health Agency of Canada, our funding agency within Quebec, Genome Quebec, as well as federal funding agency called Innovation Canada, as well as the MI4, all of which, without help from all of these partners, none of this would have been possible. And then I'll stop there to answer any questions that you may have about the work that I presented. Thank you for your time.
Thank you, Brent. This was really a great talk and really illustrate the power of international collaboration in the face of the pandemic to kind of quickly come up with a way to perform genetic analyses and identify potentially targetable pathways that are involved in our defenses against this virus. This session is open to questions. I haven't seen any questions on chat, but please feel free to raise your hand or unmute yourself and ask questions. Please don't be shy. Maybe as we are waiting, you know, I think another comment that I wanted to make, actually a couple of comments, is it's very clear from this discovery and from the progression of actually a sequence of discoveries that you described that you know, there's a great value to large population-based genomic data sets, right? Because some of the discoveries really leveraged the pQTL data sets, the splicing QTL information, and of course kind of crowdsourced genetic analyses to come up with these conclusions with the Mendelian randomization approach. And the second comment is, it's very clear your talk really highlights the value of diversity, ancestral diversity, and diverse representation of different ancestries in large-scale biobanks, and how that can help with fine mapping, putative causal variants. So that these are a couple of comments that I think really your talk nicely highlights these features.
Yeah, so I agree entirely. People may know on this call that the majority, in fact, I would say the vast majority of genomes that are available for study are from people of European ancestry, which is a really limited amount of genetic variation, and is really would be greatly enriched by having large-scale biobanks, such as what you've developed at Columbia, which really have a large proportion of representation of non-European genomes. So Christoph, I can't remember the numbers, but I remember being quite impressed with the non-European representation within the biobanks that you've contributed to this pandemic.
Yeah, so our biobank is really representative of our patient community here in Upper Manhattan, which is very diverse. It's actually the patients in the biobank, if I remember correctly, are 60% Latinx, mostly Caribbean Hispanics, predominantly from the Dominican Republic and some from Puerto Rico, and then we have 20% African American patients from Upper Harlem, and we also have some Asian patients, to a smaller East Asian, predominantly Chinese patients from Lower Manhattan, and then the minority is actually Europeans.
Yeah, so I mean, it's such a wonderful resource. And I think that targeting funding towards those resources rather than just sequencing more and more European ancestry genomes will be incredibly helpful for the field.
Yeah. I think there is a question here in the chat, actually a couple of questions. So let me start with the first one. Do you have any information about heterozygotes versus homozygotes, and I guess the effects of OAS1 locus? And do you have any data on family segregation and susceptibility? I guess if the homozygosity or heterozygosity correlates with the disease.
Yeah, so absolutely. So there's clearly an additive effect across homozygotes, heterozygotes, and homozygotes alternate alleles at the OAS1 locus. That's very clear. So the individuals who are lucky enough to have are homozygote for the G allele have much stronger protection than people who are homozygote for the A allele. We've never done any in family studies. There are clear advantages of doing so, but the population level genetics, I think, tells a fairly compelling story that there's a clear effect across heterozygote, and homozygote groups.
Okay, great. Then there's a second question about splicing variants. Were they originally detected in analysis of gene expression data, or were they predicted directly from the sequence data?
So they've actually been predicted for a very long time. So in fact, the first description of the splice site variant was described by my colleague at McGill, Jacek Majewski. I was actually giving this talk at McGill, and Jacek pointed out to me after the talk. He said, "Oh, Brent, you just found something that I published like 15 years ago," which was a little bit embarrassing because I didn't know that he had. But no, this is a clear splice site variant, which has been detected both through bulk RNA sequencing of blood, as well as single cell RNA sequencing has been shown to have clear effects upon all cells that are known to express OAS1. I'm not sure, Dallin, if that answers your question.
Okay, any other questions? All right, I guess no more questions. Feel free to contact Brent directly with questions after the talk. And thank you so much for attending the seminar. And Brent, thank you for a great talk and really, amazing progress in our understanding of these allelic effects for COVID within a really very short time frame of the pandemic.
Thank you. Yeah, thank you so much for the opportunity to be able to present. And we're looking forward to working together through the pandemic.
Okay, great. Thank you so much.
Cheers. Yeah, bye now.